Well, hi everybody. Good afternoon. Most of you know me. I'm the loudest person in the department. As you can see, I'm a fisherman, like JC is. Got that shark with him. It's 50 pounds at Assateague. Thought it would just be cool to show. But the title of my talk today is the involvement of IGF binding protein 2 in prostate cancer progression and metastasis. Now, as proposed by Paget, most cancers tend to spread to and take up residence in favorable microenvironments within the bone. Prostate cancer, being no exception, tends to spread to regional lymph nodes and bone, where it causes the formation of osteosclerotic lesions. As shown here, some that have taken up residence in the lower lumbar spine from this X-ray radiograph, as well as in this bone scan taken at UVA, where Dr. Sykes used to work. You see a great concentration of these bone lesions in the pelvic area, as well as all around the spine. The processes of prostate cancer tumorigenesis and metastasis entail multiple adaptive landscapes to which prostate cancer cells must overcome, including that of anchorage-independent growth, evasion from the immune system, increased motility and invasion, as well as what some people would call, some people would also consider the attainment of androgen-independent, or what's now called castration-resistant, tumor regrowth after androgen ablation therapy. So these processes, all the steps in these processes include changes in multiple cytokine signaling and growth factor signaling, which include members of the IGF axis, or insulin-like growth factors. The IGF axis consists of insulin-like growth factor 1 and 2, which are soluble ligands that can interact with, through differential binding affinities, they're transmembrane receptors, as shown here. IGF, when it interacts with IGF-1R, can signal for the cell to have changes in cellular survival and proliferation. Along with these members, there are also 6 high-affinity IGF binding proteins, which have differential binding affinities for the ligands themselves. By interacting with them, they can modulate the bioavailability of these ligands in the pericellular and extracellular space. In this way, they can attenuate signaling through the IGF-1R and/or potentiate it. In the Sykes lab, we predominantly focus on one of the binding proteins, IGF binding protein 2, which seems to have differential expression during prostate cancer progression, as shown here by this representative Western blot performed by Dave DeGraff using the LinCap, or the human LinCap prostate cancer progression model. As you see, the androgen-sensitive parental castrate-sensitive cell line as compared to the increasingly metastatic androgen-independent derivative cell lines. There seems to be an increase in BP2 secreted in the culture media of these cells. Multiple Western blots were analyzed through densitometry, and there seems to be a statistical significance of the levels of BP2 secreted from B4 cells as compared to LinCap. Now, along with these dynamic changes in its expression, it's also been shown to be, or have functional implications in other advanced neoplasms, such as that of advanced brain, bladder, breast, and gynecological. And specifically pertaining to prostate cancer, it was interesting that Martin Gleave in his group in 2003 used BP2 overexpressing LinCap cells and normal LinCap cells, and subsequently injected them into SCID mice and assayed for tumor volume, increases in tumor volume over a couple weeks period. And as you can see, there's 2 things to take note of here. One, the BP2 overexpressing LinCaps seem to have quicker time for tumor take, as well as after the clinically relevant point of androgen ablation, have decreased time to attain that androgen-independent, or what's now called, as I said before, castrate-resistant tumor regrowth. And this data here was mirrored and complemented by the data shown by the PSA levels monitored in the same mice. Androgen-independent, or I keep saying that, but it's now called castrate-resistant prostate cancer, it tends to be associated in patients with that also have prostate cancer-induced bone lesions. And these bone lesions have a characteristic osteoblastic phenotype. And it seems that BP2's expression in the osteoblastic prostate cancer cell lines seems to be much more dramatic, as shown here in this Western blot, also performed by Dave. Like I said, in the osteoblastic cancer cell lines, there seems to be a dramatic increase in BP2 in both the culture media and cell lysates, while compared to the osteolytic prostate cancer cell lines that have near an absence, a complete absence of BP2, as well as that of the osteolytic breast cancer cell line. So the previous functional data I alluded to, shown in the literature and in our lab, is most likely BP2 acting through IGF-dependent pathways, as I explained during the IGF axis slide. But there are also actions of the binding proteins that occur through IGF-independent pathways, including that lead to changes in transcriptional regulation and integrin binding, which in itself could lead to changes in cellular motility, adhesion, and survival. Those are all cellular behaviors that are altered during any type of cancer progression, let alone prostate cancer. So taken together, the data I've explained in the literature and from our lab, as well as some preliminary functional assays I performed on BP2 in the absence of IGF. It brings me to my hypothesis that BP2, acting through IGF-independent pathways, can stimulate the metastatic phenotype of prostate cancer and promotes the trend towards the osteoblastic phenotype in prostate cancer-induced bone lesions. To address this hypothesis, I have the following aims proposed. Starting with looking at BP2's metastatic stimulatory effects on prostate cancer, looking more into how it performs these actions in an IGF-independent manner, and the third is to actually decipher how it may be promoting or stimulating the osteoblastic characteristic of these bone lesions. Today I'm just going to primarily focus on aim 1, which I started to look at with the help of Christine Aguiar. Thanks, Christine. As we did wound healing assays in the lab, where the non-metastatic LINCAP cells were plated at confluence on 60-millimeter dishes and serum starved and then scratched with a pipette tip. Over the period of a couple days, when the cells were in the absence or presence of increasing concentrations of BP2, as shown here, the wounds were allowed to close. And as you can see, in the higher concentrations of BP2, exogenously added, you see an increased wound healing activity. This is suggesting that BP2 is possibly stimulating cellular motility in this assay. This data was complemented by invasion assays I performed, where I plated LINCAP cells, the same cells, on growth factor reduced matrigel to mimic the ECM, and allowed those cells to invade through the matrigel and through an 8-micron pore nitrocellulose membrane. And I did this while I treated the cells with increasing concentrations of BP2, either in the bottom chamber or the upper chamber of the invasion assay plates. And as you can see, there seems to be a dose-dependent increase in cellular invasion with increasing concentrations of BP2, with, as you bring your attention to panel B, a statistically significant change in cellular invasion between the higher concentration of 2 micrograms per mil. So cellular motility and invasion most of the time are associated with, if not directly require, changes in the actin cytoskeleton. I decided to, at different time points, treat the LINCAP cells with BP2 at the higher concentration in which I saw these pronounced effects, and then subsequently stain their cytoskeleton, their actin cytoskeleton, with rhodamine phalloidin. And what I saw at the 2-hour time points was that there seems to be an increased recruitment of cortical actin bundles to what seems to be an increased number of cellular processes. This effect was prolonged to the 24-hour time points, as you can see here. And previous to this, I had generated an additional tool in the lab where I had a GFP actin fusion construct, and I decided to use that to transfect into the prostate cancer cell lines, one of them being the LINCAP cell line. I decided to use that transfected line to get a more non-static, real-time understanding of what's going on the actin cytoskeleton while the cells were in the presence and absence of BP2. So this video is awesome. So, and the 2-hour control to which or the 2-hour time points I decided to choose because of the earlier effects that I saw. These are 12-minute videos, and before I even show the activity, take note on the difference in cell shape. But when I play this, you can see that there seems to be a moderate amount of activity occurring here at the proximal ends of the cellular processes in the control plate. But in the treatment plate, yeah, it's pretty cool. At the periphery of the cell, you see a great increase in cellular activity, including that of what seems to be increased filopodia and lamellopodia formation. Now, oops, sorry about that. So, increased cell motility and changes in the active cytoskeleton are oftentimes associated with focal adhesion turnover. So with the help of my undergraduate, Dylan Audette, we decided to look at activation state, levels of activated FAC within the cell, within the LNCAP cells. And we performed Western blots for the presence of FAC, phosphorylated FAC at tyrosine 397. And we saw two things. One, there seems to be an increased level of activated or intact active FAC with increasing concentrations of exogenously added BP2, as well as what seems to be an increase in an active 95 kilodalton active FAC fragment. And after statistically analyzing multiple Westerns through densitometry, it seems that there's a statistical significance between both intact compared to actin, intact FAC compared to actin, and active FAC fragment compared to actin. So with that, the suggested conclusions that I drew were that BP2 can stimulate the migration and invasion phenotype of LNCAP cells in vitro, as well as stimulating familipodia formation, and most likely through dynamic changes in FAC activation states. And some future experiments I have proposed for AIM-1 is to address BP2's actions on cellular proliferation, some additional migration assays, as well as adhesion and soft agar colony formation. And right now I'm in the middle of generating two tools in the lab, BP2 overexpressing prostate cancer cells, as well as knockdown prostate cancer cells. BP2 knockdown prostate cancer cells. And I'd like to use them to intraosseously inject into bone and then monitor for changes in PSA levels and tumor number, as well as tumor volume. And it's important to take note and reiterate that all those functional assays I just showed you guys were in the absence of IGF because they were in serum-free conditions. So that gives a lot of hope for AIM-2, which would be establishing IGF-independent actions of BP2, which I would first like to do the immediate experiments I would like to do is immunofluorescence for BP2 to assess its association with the cell surface, as well as using an IGF-1 inhibitory antibody that Erica Dashner, she's here, usually uses in Dr. Van Golen's lab to inhibit the IGF-1R activity and inhibit IGF signaling. And to refresh everyone on AIM-3 about BP2's possible role in promoting the osteoblastic characteristic of these bone lesions, I would like to, in vitro, see if BP2 can actually induce the differentiation of pre-osteoblasts, as well as using those same tools that I alluded to before for looking at, this is the intraosseous injections. So to intraosseously inject these tools, these LNCAP BP2 overexpressing lines and knockdown lines into bone and assaying for different osteoblastic and osteolytic activity. And with that, I'd like to thank everybody, Dr. Sykes, who's now Dr. Dave DeGraff, Christine for the help with the migration assays and her support, Cynthia for her help with regulation stuff that I didn't show, Dylan, his help with the FAC stuff, Erica, Faith, Noosh, Freddie, Dave, computer expert, Heather, and Keith. And also my committee members, Dr. Van Golen, Dr. Farage Carson, Dr. Mason, and Dr. Cooper. And with that, I'll take any questions, guys. Be nice. Yes.
How did you settle on those concentrations of BP2? Are they physiologically relevant?
Oh, good call. Actually, so.
Sounds like a question you were asked at your committee.
Yeah. Oh, yeah. So it's interesting because patients, it's been shown in 2 or 3 studies that serum levels of BP2 in normal adult patients are about 140 nanograms per mil. So yes, I know that's actually a 20th of the concentration I'm using. However, they seem to increase with age to 240 nanograms per mil. And in prostate cancer, they go to half the amount that I was showing. Now, yeah, I know it's still half of what I was showing. However, that's the serum concentrations. It's not known exactly how much is produced locally within the prostate, which is the most important. So if that does, hopefully that answers it.
You also didn't show any graphs for how BP2 activated the MAK. I'm just looking for statistics and curves. I didn't see that. All you said is it statistically increased.
The fact data.
Yeah.
Oh yes, yeah. I could have. Yeah, I should have shown the graphs. But yes, you're right.
So what fold stimulation?
It was 1.5. At the higher concentration.
A lot more than that from that gel.
Well, keep in mind that the actin goes down.
So it's even more,
Right,
right?
that's what I would
So,
say.
so since actin's going down, then the ratio is even going to be more.
Yeah, I think the active FAC fragment compared to actin was 1.5, and I think it was, I can't remember off the top of my head, but I think the intact FAC compared to actin was over 2. But I had an issue with trying to figure that out because at first I thought of looking at intact actin or intact FAC compared to the active FAC fragments, right? And I couldn't. I did a lot of different cross analyses between the fragments and the actin. So, but yeah, I mean, I can let you know after the talk.
We're to beat on graduate students on these things.
I know. Yes.
And it's something that I was dinking around with cameras and remotes, and you might have said. But can you kind of just briefly restate why the hypothesis is that BP2 is affecting the kind of the osteosclerotic phenotype of the MET,
Okay,
and not some
sure.
other feature of prostate
Sure.
cancer?
Actually, I thought you were going to ask that question because of Keith's thing last week. But so if you go back to
I didn't take mine.
where is it? Actually, it's
You very well may have said it. I just.
no. So the data I showed here is just expression stuff, which absolutely you're right. Doesn't indicate anything. But I also described briefly that there were other functional data out in the field that are suggesting its role in osteoblastic activity, such as DiMambro et al. In 2007 used BP2 knockdown mice and showed that they had a decrease in...
Knockdown or knockout or knock?
Oh yeah, I'm sorry, knockout.
Okay.
Sorry. And they had a significant decrease in bone mineral density, as well as...
Okay. That's really important.
It's I know, and it's also cooler that...
But the thing is that though, did you mention that? In that kind of, that's an explicit, important connection.
Well, I'll save that for the questions. Baited you in, I'm a
it.
fisherman. Anyway.
Did I give you a hard time about that just like 20 minutes?
Yeah, I think Mary just gave me a hard time about that.
Okay.
But but along with that bone mineral density stuff, they looked at a bunch of other things too, and it seems like osteoblastic activity dramatically decreased, as well as osteolytic, but not to the same extent, which would greatly hint towards that not only a predominant osteoblastic phenotype in those prostate cancer-induced bone lesions, but it also explains the osteosclerotic, which is a mix between osteolytic and osteoblastic. Okay? Oh, great.
Just as a suggestion in the future, that's so important to support your hypothesis.
Yeah.
You would have brought it out more clearly.
Absolutely. I had I thought that I had like a limited amount of time, and I don't want to.
Oh, you do a little bit of that.
Yeah. So.
So you say that this this BP2 is a player in this. How's it working? What's your hypothesis as far as how it's actually causing these problems?
Well, all right. So there's a couple. There's a lot of ways I can answer that. Right now, I'll show you my working model. So my working model right now is that BP2, when you take away IGF, this is its complete independent actions of IGF. When you take IGF out of the equation by serum starving the cells, you take out IGF signaling. And then by what I do in the lab, by adding extra BP2 to the situation, I neglected to mention before that BP2 has an RGD domain and a heparin binding domain, which through both can interact with two cell surface integrins. Its signaling through those integrins is a little hazy right now in the literature, but alpha V beta 1 and alpha 3 beta 5, alpha 3 beta 5 and alpha V beta 1. Those two integrins associate with BP2. And when I add that extra BP2 to the culture media, I think what's going on is there's an increased recruitment of focal adhesion kinase to the cytoplasmic domain of these integrins, which is probably leading to one of two things. Either increased MMP production to help in the cellular invasion I saw in the invasion assays, or, what's most likely occurring, is the recruitment and subsequent activation of Rho GTPases, leading to changes in the active cytoskeleton. And above and beyond that, that FAC fragment that I showed you guys before, it's really interesting because that is a result of calpain cleavage, which is a calcium-dependent. I know I'm getting myself into a whole other ballgame here, but might as well take a shot. Calcium-dependent.
Or get shot down.
It's a calcium, which calpain is a calcium-dependent intracellular protease that acts on FAC, and it also acts on the androgen receptor, which is very interesting. And I'll draw something up here real quick, if I could.
Okay. I'm going to have Shri cut your talk.
Okay. All right.
You can draw as she loads.
All right. Go for it.
You keep talking, she's loading.
Alpha B, V beta 1.
So
FAC.
enough to answer your question, because I have to ask the Moors if they have questions in a second.
Okay.
Give them a chance to say something.
Yes, I have a question.
Nush is going to ask you a
About
question.
20 questions.
Yes, Noosh, what's up?
Yeah, then nice seeing you.
nice hearing you.
Can you just go back to your Western blots?
Actually, you can't.
No,
You can't use the slides right now. He can. Okay, here we go.
could I just address this one real quick? Okay. So, Dr. Newman,
Actually, I was getting at the level of CMC4 cells and P-type cells. Comparing it to the first Western blot you showed where the level was higher in C4-2B, I think the second one was not consistent with the first one.
the second one, so back to, you want me to go back to Dave's data? Yeah,
I think both of it was Dave's data.
right here. You mean this one?
Yeah, that's not that exciting.
Is that what you're talking?
You quantified it, yeah, from the graph that you quantified, it looks C4-2B. So
Oh yeah, you're absolutely right. But that's why I stated that this graph is a result of multiple, I think at least 6 Western blots performed on the cell line, on this expression.
For osteoblastic cell lines.
Go back to that.
There to the slide for blastic analytic cell lines. I think this one was not.
No, you passed it out.
My bad.
Yeah,
My bad.
okay.
Oh, right here. Yes.
No, plastic
Oh,
analytic.
yeah. Sorry.
PC3, I
That's
think,
that's back at the beginning. I'm all messed up. I'm trying to answer 2 questions at the same time. Yes.
Okay, so C4 to B4 seems lower than LINCAP in condition medium here. So how do you explain if, was it previous lesson from condition medium also, or was it total?
So why is this different? Is this conditioned media versus total cells?
Yeah, so this is the culture media as CM is here for LINCAP. Yeah, and this is the cell lysate. This is the BP2 from the cell lysate of each of these cell lines. So basically what it's demonstrating is that BP2 is present in the cell lysate of these osteoblastic stimulating prostate cancer cell lines, and also present in the culture media of these osteoblastic stimulating cancer cell lines. And if your question is why is there less here compared to there, this is just one representative Western blot from multiple, and I'd have to look more into why there's actually a decrease. But just because there seems to be some present, that what is important to note is the difference between this lane and the osteolytic lanes. Okay? Yeah. So you remember that from Dave, right?
Okay.
Oh yeah. Okay. Can I get back?
Yeah, you can.
We'll talk later.